Hello, and thank you for joining this new England Biolabs presentation entitled Reagents and Technologies to Address Pandemic Response, Partnering with NEB. I am Dr. Morgan Moreau, NEB's Territory Manager for the Great East of France. After introducing our company and values, I'll highlight some of our reagents for SARS-CoV-2 detection and diagnostic developments. I'll then introduce our customized solution capabilities and how NEB can be a collaborative partner for your developments. NEB has been driving innovation for 45 years. This family-owned company was created by Don Kahn, pictured on the right. In the mid-70s, he started to commercialize the first recombinant enzymes for other researchers. Since then, he established this company as a company of scientists for scientists. Over 40% of our employees are in research and development. We are now a world leader in the discovery and production of enzymes for molecular biology applications. Our portfolio grew from restriction enzymes and cloning reagents to high-fidelity polymerase used in NGS and molecular diagnostics. We also have enzymes and master mixes for isothermal amplification and qPCR. We have a portfolio for nucleic acid extractions and a savoir-faire in NGS reagents. We also provide products around RNA biology, such as RNA synthesis and genome editing enzymes. Lastly, we have products for protein expression, since it is the core of our savoir-faire, as well as reagents for glycomics. We are mostly renowned for the high quality of our products and unprecedented technical support, thanks to the direct contact with our R&D teams. So now that you've got an overview of who NEB is, I'd like to present our core products for diagnostic development. We have solutions for nucleic acid extractions with unique specificities, but given the time constraints here, I choose to focus on our virus detection, sequencing, and epidemiological tools. First, a technology that has gained a lot of momentum during this epidemic is loop-mediated isothermal amplification. The amplification here takes place at a constant temperature, so you just need a heating block. It works with a set of 4 to 6 primers and a strand-displacing polymerase. The design of those primers is critical to the success of the reactions. So to help researchers, NEB developed a user-friendly online tool to help you with the design of the primers. The reaction generates long concatenators and such massive amounts of DNA that numerous methods of detections are possible. You can either use real-time fluorescence or endpoint fluorescence, lateral flow detection, agarose gel, or turbidity. You can even use a simple visual detection method or a simple plate reader. The colorimetric lamp is unique to NEB, and it uses the release of protons during amplification to induce a color change with the change of pH in the reaction. Interestingly, this technique has been widely used on direct patient samples, shortening drastically the time from sampling to result. We've had these products in the catalog for a long time. They were mostly used for veterinary application, as it is particularly suited to screen large herds in the field, or to use at the point of care diagnostic in the field. The current crisis really shed light on its use as a potential new way to do diagnostics for human health, simply because it doesn't require specific equipment and that the readout is available in 30 minutes. Since we are interested in providing more diagnostic tools to the community, we have developed a multiplex method for LAMP to increase the throughput of this technology and detect multiple pathogens in a single reaction. In this example, we detect 4 targets at once with a real-time or endpoint fluorescence readout. So the take-home message for the LAMP technology is that it's so simple to run that it can be deployed for mass screening. It has been already included in many diagnostic applications developed by highly renowned institutes such as Harvard or MIT. These reagents are included in kits that have been approved by the FDA or the CNR, or that are included in CE-IVD kits. Nevertheless, qPCR stays the gold standard for SARS-CoV-2 detection and other diagnostics. Since NEB is fairly new in the qPCR field, I'd like to introduce to you our Lunar Portfolio. The data presented here show a comparison of our one-step Lunar RT-qPCR probe master mix to other suppliers' reagents. The data were obtained based on selection criteria coming from the MIKE guidelines. This product has been out for 3 years now, and these performances have been since confirmed by many of our users. It is a very robust master mix as it's been shown to be compatible with direct saliva and nasopharyngeal samples without extraction, such as in the FDA-approved saliva direct protocol. There is currently 5 FDA-approved kits, including Lunar reagents. And we now have a 4X master mix with UDG for increased multiplexing and carryover prevention. Another example of the robustness of Lunar reagents is that results are consistent no matter how long the plate sits at room temperature before the run starts, which is not the case with the TaqPass reagents. And this is possible thanks to our dual enzyme control. That's innovations from NEB. We will soon release a SARS-CoV-2 Lunar qPCR kit for research use only, as NEB doesn't intend to develop diagnostics, but is happy to provide performant products for people to develop their own kits. The last group of reagents I'd like to introduce is our library prep reagents. NEB provided Solexa Illumina with enzyme for their first library prep kits, so NGS reagents are totally part of NEB savoir-faire. I'd like to highlight here in this very crowded slide some RT-qPCR application. RT-qPCR protocols were extensively shared during the first wave, and they were updated regularly with new improvements. Our LunaScript reverse transcriptase took over the renowned superscript 3 in the RT-qPCR protocol, and our NGS reagents for library prep have been used widely by many reputable institutes, including the Sanger Institute, Pasteur Institute, MIT, or Harvard, and even the CDC, to provide the most accurate sequences of SARS-CoV-2. I invite you to go and have a look at our scientific poster on the 4ID website showcasing the performance of our soon-to-be released NEB Next RT-qPCR kit. Now I'd like to present how NEB can be your partner for your product development. As I said, it's not our intention to develop diagnostics, but our customers are. And the current crisis really highlighted the role we can play in helping them developing the right product throughout its life cycle. First, we work closely with our clients' research and development departments at the early stage of the project to provide the best product and technical advice. We are then capable of manufacturing large quantities to solidify your supply chain, adapt formulation if needed, as well as packaging and kitting. We can even put your logo on our products. We maintain the appropriate ISO certificates such as ISO 901 and 13485 for regulatory needs, and we have a new GMP grade production facility too. We can continue to help you scaling up while minimizing cost. We've played a role in molecular diagnostics since 2010 and in advanced therapeutics since our new GMP grade facility opened in 2020, producing reagents useful for nucleic acid-based therapeutics. Finally, you'll find all of our COVID-19 related resources, such as product, protocols, and citation, on international.neb.com. Just type in supporting COVID and you'll find all our COVID related resources. And don't hesitate to reach out to me or to Nicolas Doll, our NGS and OEM bulk representative, who is also attending this Congress.